Okay, good morning, everyone, or good midday. I don't know what to call it. It's not quite afternoon yet, not quite morning, but welcome to the first workshop of the Metabolomics meeting here in Niagara Falls. Thank you for choosing the ion mobility workshop. What we're going to do today is discuss some different workflows and resources that are available for doing metabolomics with ion mobility mass spectrometry. So the sort of order of the day today, I'm going to give a little brief introduction into ion mobility and talk about some resources that we're not going to have presentations on from the others. And then we're going to flip the order here a little bit. So then Tomáš Pluskal from the Institute for Organic and Chemical Biology in Prague will talk about MZmine 3. And then Jess, Bade, and Chang from PNNL will talk about their new program, DEIMoS. And then we'll sort of come back to me at the end for a little bit to talk about methods for collision cross-section predictions and resources for where you can find CCS values for metabolites. All right, so to get everyone on the same page, because you all might be coming from sort of different backgrounds of ion mobility experience, there are sort of three main types that I think of as like the gas phase classical style of ion mobility. So the original one really is this drift tube style ion mobility instrument. These were the ones that were sort of home-built instruments that were, you know, the first that were used with mass spectrometry and have been around the longest. And so these have a couple different advantages compared to the others. And so one is that you can do direct CCS measurements from drift tube ion mobility instruments. What else? And it's sort of a comprehensive method, meaning you get a range of drift times across the whole experiment. So you're not doing any sort of scanning or selection in the ion mobility dimension. You're doing a full separation in that sense. There are now commercial versions of these drift tube ion mobility instruments, the first being the Agilent 6560, which is attached to a QTOF. But there are a couple other smaller vendors that sell these now as well. The next one is the traveling wave style ion mobility instrument, and this was actually the very first style of commercially available ion mobility platform that was out there. These are the Waters Synapse. And now there's a couple different variety of those. So there's the cyclic traveling wave instrument from Waters, and now also the mobile ion SLIM technologies also use traveling wave. And so these differ from the drift tube instruments in that you have to do a calibration process in order to obtain collision cross-section values. And so that means that you're using a drift tube value that is somewhere in the literature or in an online database, and you're going to measure that same compound, get the drift time, and then calibrate that to the drift tube CCS value. In all other sense, it gives you basically the same information as a drift tube ion mobility instrument. There's just sort of that calibration process to be aware of, and they work very similarly. The third style of ion mobility is the trapped ion mobility method, which is really quite distinct from the other two. It's sort of a backwards way of doing ion mobility. And so in the first two, where you would have normally your smallest sized, ions coming first through the drift tube, trapped is the opposite. So you're holding the ions in this trapping area, and you're slowly letting them come out by dropping down the field in the trapping region. And so what you actually get are the larger ions coming through the trapped mobility first, then the smallest. This also needs to do a calibration process in order to get collision cross-section values. The process is about the same as what you do on the traveling wave ion mobility instruments. And I just want to say both, or all three of these, right, what we're talking about with these instruments is using nitrogen as the drift gas. So if you change the gas, there's going to be some change in your CCS value. So just make note, like what we're going to talk about today, I think pretty much exclusively will be using nitrogen drift gas, and so nitrogen CCS values. Oops. Okay, so in all of those instruments, regardless of what type of ion mobility platform you're using, the general arrangement of where the mobility cell is pretty consistent amongst all of them. So typically, this is going to be something that sits after your ionization source because it's ion mobility, right? So it has to be an ionized analyte for this process to work. And it sits before the mass analyzer. And so what's really nice here is that you can couple this to MALDI, you can couple this to chromatography, any other sort of like front-end separation you'd like to do. So it could be supercritical fluid chromatography. Those are all potential upfront separations that you could incorporate and still have ion mobility data. So the nice property and the reason why we all want to use ion mobility and have these CCS values is that it gives us information about the size of the molecule. And so this particular slide, think in your mind, trapped ion mobility would be backwards. This is more for like drift tube and traveling wave. But depending on the order, the way that the ions elute and their drift time, you can use that information to get something about the structure of the ion. And so in this particular case, these could be exactly the same mass to charge. They could be exactly the same mass, so same chemical formula, but because their shapes and sizes are different, they would have different drift times, and those would give us different collision cross-section values. So when you combine that with the mass spectrometry information, the mass to charge information, you can have these really lovely organized representations of your data in two-dimensional space where you see these trend lines for the different types of biomolecules. And this is because that there are certain inter and intramolecular interactions in the molecules that give them their shape. There are certain sort of building blocks that they all have in common, right? So there are only a certain number of peptides, and proteins only expand in a certain dimension, and they have a particular shape, right? Same thing for lipids. They have fatty acyl tails that elong in sort of one direction, and they have head groups that can swap out. So there are some sort of shared structural properties within each of these biochemical classes that determines their relationship between size and mass. And so that's one thing that's really important to think of, is like there's this common idea that ion mobility is orthogonal to mass spectrometry, and it's not. There's actually a relationship between size and mass, right? So there's, to some extent, you would expect the size of something to scale as its mass goes up. Now, the other lovely thing about ion mobility is that it really is the perfect time scale to fit into an LC-MS experiment. And it doesn't necessarily add a lot more time. So if you think about the dimensions and the time scales of what you're working on, if you're doing chromatography, right, your sample, you're going to have 1 chromatogram per sample. It might take you 20 minutes to run that separation. The mass filter is operating on a range of milliseconds, right, hundreds of milliseconds. The ion mobility dimension is 10 milliseconds to maybe a little bit longer if you're doing higher resolution. So maybe you're still in the millisecond range, but maybe you're getting more to like 50 milliseconds or so. And then the time of flight, the mass analyzer is working the fastest. So you have this perfectly nested sort of Russian doll time scale setup so that for every chromatographic peak, you have multiple ion mobility experiments happening. You have many individual mass spectra being collected across that ion mobility experiment. So generally speaking, some of the advantages for adding ion mobility into your metabolomics workflows are places where it's beneficial to have that structural separation. So the biggest one, I think, affects anyone who's trying to do metabolomics is the isomer problem. So there are many small molecules that have exactly the same chemical formula, but have different structures and have very different biological importance, right? So we want to be able to separate them so that you can accurately monitor all of those metabolites individually. The other benefit is that you can use the CCS value as a way to filter potential candidates during the annotation process for your metabolomics experiment. And finally, one of my favorites is that it really improves the cleanliness, the quality of your spectra if you're doing data-independent MSMS acquisition, because we have another dimension there to separate things out, and you can actually use the drift time information to select out just the fragments from the precursor that you're interested in. And so the impact really is that you have an opportunity to reduce or potentially even eliminate the chromatographic separations, because it becomes less important to perfectly resolve every single analyte in your chromatogram. So we're going to talk a little bit about some examples that cover each of these. So this is a great example of the isomer issue. So these are constitutional isomers. They have the same chemical formula, but the structures are different because the functional groups are connected in different places on the molecule. And so on a typical drift tube ion mobility instrument like the 6560, working at a resolution around 60, you can partially separate these different molecules at certain adducts. So you can see that when they're just the protonated adduct, you get pretty nice separation of the A35DP and the DGDP, and the A5DP sort of sits in the middle. So if you had all three of these, you maybe wouldn't be able to truly separate them, but if you had a mixture of two, perhaps you could. As you start looking at different adducts, so you're adding a sodium onto that adds a little bit of size, that gives you a bit more of a handle on being able to resolve them. But as you move into negative ionization mode, then it becomes a bit more challenging. So there's a lot of nuance to being able to do these kinds of higher resolution separations of isomers. It depends quite a lot on the analyte you're looking at, the adduct that you're looking at, and obviously your instrument. So there's more types of isomers than just constitutional isomers, and those aren't necessarily even the hardest ones, right? So the harder ones are the stereoisomers. We're talking about like enantiomers, diastereomers, and those are still really a big challenge for ion mobility in terms of sort of the medium range resolution instruments. So like at a resolving power of 60, there are certain things that you're going to be able to separate. There are going to be some that you just will not. So even for the constitutional isomers, these are all 11 different isomers of leucine, so it's very small metabolite. So they're changing in how the structure is branched and where the functional groups are. 3 of these constitutional isomers you should be able to separate at a resolving power of about 60. But another pair of constitutional isomers, so leucine, isoleucine, sort of the big ones that we're all most familiar with, would be pretty challenging, or impossible, I would say. If you had a mixture of these two, right, in the black trace, you see just one peak. And as you start moving to more subtle differences, like diastereomers, alloleucine and isoleucine, and the enantiomers, so L versus D, those you're unable to separate at this particular resolving power. So a really nice example of what you can and can't achieve on, let's say, like a drift tube instrument with 60 resolving power is in this study from the McLean lab back in 2018. And so they were looking at the, what is it called, the MSMLS. It's like a big library of metabolite standards, and they're all in different wells, so you can look at them and measure them individually. And they did a nice analysis of all the different isomers that were in that collection. What was the difference in the CCS value between the isomers, and what type of resolution or resolving power would it actually take to be able to separate them? And so you can see that there are quite a few that are in this sort of nice sweet spot between 9 and I think it's hard, 2% different in CCS between the isomers, that if you have a resolving power around 70, you could be able to separate those on your instrument. But at the same time, there's quite a lot of isomers in this panel that have very, very tiny differences in CCS. And so that would require resolving powers upwards of 300 to be able to distinguish them. So looking at this in a different way, right, we can kind of predict where we would need to be on this line of resolving power in the ion mobility dimension to resolve particular types of isomers. And so while we can do constitutional isomers at pretty reasonable resolving powers, the higher resolving powers are really, really crucial for doing things like the enantiomers, and particularly depending on the amount of separation you're trying to achieve. So if you're trying to achieve baseline, 90% separation, it's going to take a really high-resolution, sophisticated ion mobility instrument to do that. And they do exist now. So there are platforms like the cyclic ion mobility, the SLIM device, the TIMS also can achieve pretty high resolution, and the 6560 drift tube now with a post-processing method can give you pretty nice resolving powers. Not 500, but maybe more on the order of like 200. Now, the other benefit that I mentioned for adding the ion mobility is this candidate reduction and filtering process. So if you have data, in this particular example, this was data that was collected in Facundo Fernandez's group, and they did the same experiment on an Orbitrap setup and a Waters Synapse traveling wave instrument, so the same chromatography, same samples. They had tandem MS data from the Orbitrap system that they used with Sirius 4 to get some potential identifications for 19 of these features that were in their data set. And so what this plot shows you really nicely is that the size of this circle tells you about how many different candidate identifications came back from Sirius. The color of the circle tells you what percentage reduction in the number of potential candidates you achieve by adding in the ion mobility dimension. And so on average, they were able to see a 28% reduction in the number of candidates, right, so potential identifications for the molecules they were looking at by including the CCS value. And so that's an average. And you can see by looking at the plot that it really covers a pretty big range. There are some that have very small improvement, maybe 3%. There are others that are a little bit better and go more towards 40 to 50% in it. So it's very much determined by the analyte itself and how much overlap there is in some of these potential molecules that you're looking for. And so last benefit here is the structure of the ion mobility setups in these instruments. So most of them include a way to do fragmentation, collision-induced dissociation, after everything has been separated in the drift cell or by mobility. So whether it's the drift tube instrument, traveling wave, or the trapped ion mobility, the collision cell is usually after the ion mobility separation. And so because the mobility part is the slow step and the fragmentation and the mass analysis are much faster, the fragments all end up having the same drift time as the precursor. And so when you look at your data, you get something that looks like this. This is a lipid example, so forgive me. This would be the drift time of all of the intact lipids, whether you're looking at MS1 or MS2. But in the MS2 data, all of the fragments then occur within the same band of drift times as their precursors. And so you can go into your data and say, I need to just look at the range of mobilities that's centered around where these were before, and you can excise out this region and really remove, essentially, the noise away from your signal and get a really beautiful, clean fragmentation spectrum. So it gives you a lot of confidence and much cleaner MSMS data, which then makes coming up with identifications a bit easier. And so another example here of how that can improve things like your chromatography. So if you've got a lot of stuff that's sort of co-eluting, overlapping peaks that may be very different intensities, you can use this sort of ion mobility filtering, even in the MS1 dimension, to sort of remove the noise away from what you're really interested in. And so you can see how dramatically it changes these extracted ion chromatograms by applying an ion mobility filtering step. Okay, so some of the challenges, though, that we face, because there's always things that can be better. One is the data analysis. So you're adding an extra dimension into your data, which means there's more complexity there in the data analysis. And while there have been some pretty good vendor sort of specific options that have come out and things that are like put out by the vendors themselves, so proprietary software, it's only been in, maybe like the past 5 years that there's some really good vendor neutral free software tools that you can use to do data analysis on ion mobility mass spectrometry data. The other thing to consider is like how the ion mobility dimension is being used in peak picking, and a lot of this is still mostly driven by there being an LC dimension. So that's something to kind of keep in mind. The second challenge that we sort of as an ion mobility community are still dealing with is the agreement in CCS values. And so there's variation from one lab to another, even if you're all using the same instrument. There can be a lot of variability between two different types of instruments. And then there's also some variability between experimental CCS values and predicted CCS values that we'll talk about. So it's important to know when you're trying to look at your data and match it to something that's in a paper from someone else's group or in a database, sort of what are the reasonable limits or thresholds that you should consider for assigning something as an actual mass using the CCS value. So I'm going to cover quickly a couple of software options that we're not going to have presentations on today. So these are, I think, 4 of the ones that I am aware of that are pretty much usable for anyone who's doing ion mobility, meaning if you're working with an Agilent instrument, a Waters traveling wave instrument, trapped ion mobility, you should be able to use any one of these 4 platforms to do your data analysis. I haven't necessarily tried them all myself for all of that, but in theory they should work. So we're talking about Skyline and MS Dial, and then Tomáš will talk about MZmine 3, and Christine and Jess will talk about DEIMoS. So you can see this nice table from Dylan Ross in this molecules paper coming out of PNNL really nicely summarizes all the benefits for these for different setups. And so they cover both untargeted and targeted experiment styles. Some have the capability to look at data-independent acquisition of MSMS. Others include DDA as well. Some have built-in libraries that allow you to sort of do the identification process within the software. Others do not. So Skyline is really nice if you're trying to find particular analytes in your data. So if you have, a set of metabolites that you should be seeing in your experiment, you can program Skyline to specifically extract out that information. And so in that sense, it needs to be used in a targeted way, but because it's very simple to work with, there are groups who are sort of using this in a like very large scale targeted manner that you put in a lot of potential metabolites that you might see and use it to pull out that data. So you don't necessarily have to know everything, but you can sort of program it in that sense. So all you need to do is have some information about the chemical formula, and it will calculate the mass to charge, the different adducts you might see, and extract out in your data all of these traces for those targets. So because this was originally built as sort of a proteomics, targeted proteomics MRM method, everything is built around having these sort of like targeted lists of precursors and product ions. And so it will extract out all of those, and then it will look into the ion mobility dimension to find those peaks. So there's some really great tutorials on how to use Skyline for small molecules and how to use Skyline with ion mobility, and so they're all on the Skyline website. You can find them. They're really great. There's videos, I think, that go with most of them. But just the general process, if you wanted to incorporate this yourselves, is to set up this ion mobility filtering library in Skyline. And so you would import your data, identify the peak bounds in the LC dimension for your analytes, and then it would build the IM library based on those peak bounds in the chromatographic dimension. So it would say, like, within this chromatographic peak, here's the drift time where that particular, mass to charge and its fragments are appearing, and add that in. And then once that library is built, you can use it on the other data files in your experiment. The other example, which is better for untargeted analysis, is MS-Dial4. And so MS-Dial4 has been around for lipidomics and metabolomics for quite a bit, and it recently added in the ion mobility step. One of the things to consider with this is that you may need to go through one or more file conversion steps before you can use MS-Dial. So it just depends on your particular platform that you're working with. You may need to go into MZML and then go into their IBF format, but it's not too bad. But here what they're doing is kind of similar to how the Skyline process is working, is it's identifying the chromatographic peaks first and then sort of expanding that into the ion mobility dimension. And so this example here is showing that at one particular retention time, when they look then in the mobility dimension, there's actually 2 mobility peaks, and so then that gets sort of split into 2 features that have 2 different drift times. The interface for working with MS-Dial is really nice, so you can set it up to do CCS calibration if you are working with a platform that requires that. There's lots of different tolerances that you can try out for how strict you want to be in the matching process for CCS values, or how strict you want to be in aligning different drift times. And then they also do have a really nice metabolite CCS library that covers both positive and negative ionization mode, and includes some predicted CCS values as well as the SMILES and INCHI keys, and also includes some ontologies. When you do this experiment, not only would you get like potential metabolite identifications based on the CCS value, but you can get some information about like what pathways they fall into. Okay. So at this point, I'm going to turn it over to Tomáš. If there are any quick questions while he gets set up and while we switch things over, we can do that.
So thanks very much. It's well known that there's always a bit of a sensitivity hit when using ion mobility, and I wondered firstly what your experience is on which versions give the least sensitivity hit, but also what implications this has for your DEIMoS MS, because obviously how much of the cleanup is actually because you're just not able to fragment as many peaks?
Yeah, so you do have a sensitivity loss. I think in our data, it's usually like an order of magnitude, and I try to think of it as what you're actually getting out is an improvement in signal to noise, even though you're taking a sensitivity hit. So it's not a like net loss in that sense, because the background you can remove away from your signals. I have not worked with all of these different platforms, and so I'm not qualified to say which one is better or worse in that sense. But yeah, you'll see if you have an instrument and you turn on the mobility, you'll see the signals come down. But I think when you really start digging in, it's that improvement in signal to noise that's the benefit there.
So sorry, just to follow up with that. I suppose the question is there anything we can do to improve that sensitivity hit? And in particular, we're very familiar with matrix effects with mass spectrometry, but I never hear this mentioned with CCS. Are there any things that affect the sensitivity that we do have control over?
I have to think about that for a bit. Yeah, and then you can see maybe our other presenters have some suggestions. But yeah, that's a little bit harder because I think the same things that you deal with mass spectrometry are going to be some of the same challenges with the ion mobility, and how you would specifically improve it in just the ion mobility dimension, not certain on. Okay. All right, so up next we have Tomáš Pluskal who's going to present on MZmine for us. If you want a laser pointer, you can use this.
I'll try to do that. Okay, the slides. Perfect. Good. So, as you heard, my name is Tomáš Pluskal, and I'm at the IOCB Prague. And I've been working on this MZmine platform for almost 20 years now. And I included pictures of Stefan Heukeroth and Robin Schmidt on this slide because they are now the key developers who have done most of the work on the software and also prepared actually most of these slides that I'm going to show you. So they deserve the credit. Okay. Now I suppose you have heard of MZmine at some point. So I'll just very briefly recap the history of the software. This platform was actually started back in 2004 in Matej Oresic's lab. At that time, they called it MASO, but luckily they changed the name to MZmine very soon. And the first paper about MZmine was published in 2006, at which point I also joined the project and I started reworking the whole platform from scratch. And we published the result of this effort in 2010, which at that point the software became kind of modular and more suitable for high-resolution mass spectrometry analysis. And then there was a lot of development, many versions followed up until we decided to introduce the next generation, which was last year, and then the paper about the third generation was published quite recently, a few months ago. And this included a lot of new improvements and new ways how to process data and many different modalities of mass spec data, which I will talk about today. So the software has now become more like a community project, although the core of the development is done in essentially two labs, in my lab and in Münster. There have been many contributions from all around the world. We have had almost 60 different developers joining the project. The project has over 92,000 downloads over the last 5 years. And on the timeline in the bottom, you can see the number of lines of code as they were increasing over time with the introduction of different modules and functionalities. Okay, so it's a platform with quite a bit of history. And in the third version of MZmine, one of our focus areas was also performance. Because MZmine 2 was nicely usable for smaller studies, couple of tens of samples, but nowadays people do much larger studies, hundreds of samples, even thousands of samples. And also with the introduction of new technologies like ion mobility, you get a lot more spectra, the data gets bigger, and we don't want the software to choke on it. So here you can see the huge improvement in throughput and speed of data processing in MZmine 3. Now that's not the only thing that's new in MZmine 3. Besides the performance improvements, we have had a lot of development in the molecular networking workflows, which are done in collaboration with Peter Dorstein's lab with the GNPS platform. But that I'm not going to talk about much today because it's not really related to ion mobility. Ion mobility is one of the main new features in MZmine 3, so you can now load, visualize, and process your data that include ion mobility dimension, and that also includes imaging mass spectrometry data, which was not included in MZmine 2. And another main improvement is the processing wizard, because many people in MZmine 2 struggled with optimizing and setting the parameter values correctly. We now have this nice graphical wizard where you essentially set a couple of main values, like what is the type of your mass spec, what is the type of your chromatography, and so on. And then it generates a batch of processing steps for you, and you can just click and everything is done automatically. It's very nice. And if you start MZmine 3, you will also notice the whole graphical user interface has changed, and it's now much more smooth, it's skinnable, it's responsive, it's amazing, and in my view, the coolest feature is this dark mode. It's optional, you don't have to use it, and you will see that in some of my slides, it's in the dark mode, and some of them is in the light mode, depending on who prepared the slide. But I'll just leave this there for a few more seconds so you can admire the beauty, okay? Okay, so as you have already heard from the first presentation, ion mobility is yet another way how to separate your ions in the gas phase. And with the introduction of this technology and also recently more and more popular imaging technologies like MALDI-MS, we get quite a diversity in the types of the data that people use and people collect. And all of these need their specific data processing strategy. So if I would summarize the biggest improvement that we have now in MZmine 3, it would be the complex support for all these different types of data. Okay. So to be more specific, if we talk about ion mobility combined with chromatography and mass spectrometry, so like in this example, LC-IMS-MS, we have mass spectra, which are collected both in the time dimension and in the ion mobility dimension. Okay? And from these, we can produce extracted ion chromatograms, which give us some peaks, and we want to detect those peaks. But for example, in the mass spec imaging processing, it's a totally different story. There is no time dimension. We may have ion mobility, so we may have the mobility dimension, and we have spectra. And from these spectra, we can generate extracted images, so we can show the spatial distribution of each mass, but it's a totally different type of data. So first, I will focus on processing the LC-IMS-MS data in MZmine. So first, about the terminology. On the left side, you can see mass spectra, so we have several ions there, M over Z over intensity, and these might have different mobility values in the mobility dimension. On the right side, you see a chromatogram, so we have retention time over intensity. We have some ions there that might be overlapping in the retention time. Again, they might have different mobility values. It's not so easy to imagine how this data actually looks like, and because you have the time dimension, you have the mobility dimension, you have the mass dimension, and you have the intensity dimension. And we humans are not very good at imagining four-dimensional data. Plus, in MZmine, we always try to use graphical previews when you're doing your data processing. So in order to see what's actually happening, we have to somehow wrap it up into no more than 3 dimensions. So if you separate the first 3 ions in the spectrum into the mobility dimension, you can show that on a 2D plot with the signal intensity indicated by color. And this is what we call the frame heat map. So frame is when you take all the spectra that are separated by an ion mobility dimension by 1 ion mobility separation, and you collapse them together. This is what we call a frame. So when we show that in 2D, mass over the mobility, it's called a frame heat map. Now, if we take the chromatographic dimension and we plot that against the mobility, we get different type of trace, again in 2D with the colors indicating the intensity. And this is what we call the ion mobility trace. So here we have no mass dimension on the right side. So these molecules might actually have different mass, but we show them separated by the mobility. So in order to see your ion mobility data, you actually have to start thinking in these multiple different ways how to look at it, because it's not possible to look at the 4D data set. So in MZmine, we have the main module for visualizing ion mobility data that we call IMS raw data overview. And there you can load your chromatogram. So here we have retention time over intensity. And that you can also configure. So you can show different mass traces, you can visualize particular mobility ranges and so on. And then in each time point in the chromatogram, you can show the frame. That means all the ions that were detected at that retention time, no matter what their mobility values are. And you can show that frame separated into the mobility dimension as the frame heat map. Now on the side of the frame heat map, you can also see a mobilogram, which is simply the intensity plotted over the mobility dimension at that particular retention time that's selected. Then you can also show the ion mobility trace, which is the mobility dimension across the retention time dimension. So essentially the chromatogram expanded into the mobility dimension. And if you select a point in that ion mobility trace, then you can show the mass spectrum at that point, which is the individual mobility scan. So this is not a frame, but this is within the frame, the individual mobility scan. And of course, you can iterate and go through these scans. So as you can see, this is quite complex. And this is just still looking at the data. But MZmine now has all these options, and they're all interactive. So you can zoom, you can click through them, and it allows you to see the data in all its complexity. Now next, what we need to do is feature detection. So this is the general workflow for feature detection in LC, IMS, MS data. Again, it is quite complex, but these figures actually come from a manuscript that we are now just finalizing for submission to Nature Protocols. So if everything works well, hopefully in a few months, you will be able to download a very detailed step-by-step protocol that should include the LC, IMS, MS, and imaging MS, IMS, MS, and also gas chromatography MS processing in MZmine. Yeah. So this will be like a step-by-step tutorial. And I think what I want to focus on here is just the topics that are relevant to ion mobility. So we can focus on the basics of the feature detection. And that means first you have to load your data in. You have to pre-process your spectra, that means detect the individual ions, remove noise, and so on. And then find your peaks, and then align the peaks into an aligned, what we call a feature list. Now, if you include ion mobility, that means you have to add an extra step there, and that's what we call ion mobility expand. IMS expanding, yes. So you have to expand your features into the ion mobility dimension. And then once you have done the feature detection, then the follow-up steps, which might be the annotation of your compounds or molecular networking, export of your data, that all remains the same. So this shows how the whole workflow is organized in a sort of plug-and-play manner. And then so if you do all this, you get your feature list in which you can see all the information that you detected in one table. So you get the chromatographic shapes, you get the mass values, you get the retention times, you get the mobility values, everything in one place. And so I was asked to give a little tutorial style presentation. So now I want to show you these steps, how they actually happen in MZmine. Yeah? So you first have to import your raw data. MZmine supports many different file formats, but if you want to include the ion mobility data, then we currently only support MZML, which can come from many different vendors, or the Bruker TeamStuff files. Okay. So you can load these by, most easy way is just by drag and dropping them into the MZmine window. And then next, you do the spectral preprocessing. And then this, if you have MZML data, the important thing is that you don't have the information about the frame. So you only have the individual mobility scans from all the mobility separations. So then you select, you want to do your mass detection and detecting all the ions in the mobility scans. If you have Bruker data, then you also have frames. So then you have to do this on all the available scans. And then if you're starting with the MZML data, then you have to construct the frame spectra. So for that, we have a module that's called mobility scan merging. So you take all the individual mobility scans in one ion mobility separation, you merge them, you create the frame spectra. And then you follow that up, because you now have the frame, so now you can follow up with the standard workflow for feature detection that we have in MZmine, which means you create chromatograms, you use the resolving module to separate your chromatograms into individual peaks, and this is something that I'm not going to describe in detail because this has been the same for many, many years, and it's not specific to ion mobility. But now imagine you detected your features in the retention time dimension. So on the top panel, you see your feature list, which has only the time dimension plot. And so you can see that you have your features, but they are not separated by the mobility. So that's where the IMS expanding module comes, and you essentially expand this in a similar way as we detect chromatograms, we detect the mobility traces. So after running the IMS expander, in the bottom part you see the same feature list, but now it has the mobilogram. So now it has the separation across the mobility dimension. Now in the very first trace of the mobilogram, you can see there are actually two peaks. So that means we have some features that have the same mass, the same retention time, but they actually form two peaks in the mobility dimension. So probably some isomers. So for that, we have the resolving module. Optionally, you can also do smoothing on the mobility trace in case the data is a little noisy. That might help, but it's optional. And then we use the resolving module. MZmine for all these resolving algorithms has several options what you can do. But so far we have found this local minimal search is the most robust, and we mostly use this module. That's a module that essentially just searches for local minima in your traces. So if you have some peaks, it will search for local minima and try to separate those traces in those local minima points. So you run this resolving, and then you end up with individual mobility peaks in your mobility dimension as well as in your chromatographic dimension. So the final outcome of the process should look like this, where you have your feature table, and for each feature you have the mass, you have the retention time, you have the trace of the feature in the chromatographic dimension, you have the trace of the feature in the mobility dimension, and there should be, in each trace should be only one peak. So we should not have situations where you have multiple peaks that were not resolved. So now the second section is about imaging IMS-MS data. So imaging, again, is a new feature in MZmine 3. You can load your imaging data, which would be typically MALDI MS, and you can visualize it, you can play with it. So you can load the imaging data, you can do extracted images by some mass ranges, you can show the spectra at individual points, and so on. You can play with it in a similar way that you can play with the ion mobility raw data. Again, we have a workflow for the feature detection in imaging data. Now the feature here means something a little different because we don't have the time separation, so we are not looking for peaks, but we are looking for extracted images of specific masses. So we have the module that's indicated by number 3, I believe, the image builder, which is the module that goes through the data, finds all the masses that are present, and builds the extracted images. And then the pink part that follows, that's the ion mobility specific part, where you separate your images by the mobility dimension into individual extracted images for individual peaks in the mobilograms. So again, in a step-by-step manner, what that means is you first run this image builder, you set the parameters, and you end up with a feature list or the image list that's shown on the right, where you have the spatial distribution of each mass that you detected. Now at this point, we are not yet considering the mobility dimension. Now in the next step, you run the IMS expander module, which is the same one that we run in the LC IMS data processing. And you get in your feature table additional columns, which then show the trace of each mass in the mobility dimension. And you might notice in this particular trace that I'm showing, there are actually 2 peaks. So there are 2 isomers in the same spot on the image with the same mass. And so we can next use the resolving module, which will turn those mobilograms where we have 2 different features into 2 individual features. So now we have in the imaging data extracted images with different spatial distribution based on the mobility values. And this is how, again, the final result should look like. So from your imaging data processing, you will get signals where you have your mass, you have the trace of that feature in the mobility dimension, and the spatial distribution in the form of the image. Now, what is nice in MZmine 3 is because it can load all these different data types, is that you can also combine them. And this is what we call multimodal data processing or alignment. And this is the feature that was highlighted mainly in the paper that we published a few months ago. So why would you want to do this? And so if you think about multi-mass spectrometry, you get the spatial distribution of your metabolites, which might have the biological relevance or the biological context, but you don't get any MS information. So it's hard to annotate your compound. You might do some MS scans in a targeted way manually, but it's very slow and you don't get much MS coverage. On the other hand, in LCMS, you get very high MS coverage, and you get lots of information that you can use for annotating your metabolites, retention time, MS spectra, comparison with internal standard, and so on. So we would like to combine the benefits of these 2 worlds. So I'll show you this on an example here, where we analyzed the brain tissue, and this was developed by Stefan, so I'm going to use Stefan's brain here. And so we take the brain slices, we analyze them by LC IMS MS, which we get data. We support, as I said, MZML or the vendor format, and also analyze the same slices by MALDI MS. So again, we get IMZML, which is the imaging format, or the vendor format. We get different types of data, so we get the spectra resolved in the time dimension for the LCMS data. We get the image distribution in case of the imaging data. And both of these can be resolved by the ion mobility. So you can see that on the very right side, the peaks in the LCMS data are resolved by ion mobility, and also the images are resolved by ion mobility. Now, the fact that we have accurate mass measurements and we also have the mobility measurement means that we have 2 orthogonal values for each feature, and that's enough to align them. So although we cannot use retention time for alignment because the imaging data doesn't have any retention time, we have the mobility value. And so we can align these data sets by the mass and the mobility value, and then you get a feature list that looks like this. So we have the extracted images, that's from the imaging data. On the right side, you can see the values from the chromatography, where we have the mass and the retention time and the mobility. And the fact that you have all this information together means that now you can annotate your compounds based on, for example, the MS spectra and the data that you have from the chromatography, and you can assign the identities. So if I highlight here, these are lipids, so we have the annotations from the lipid annotation module that tells you that the first one is PC34.1 and the second one is PC34.0. And we have the spatial distribution of these lipids from the imaging, which normally would be very hard to obtain with this level of annotation and information. What this also allows you to do is, for example, do things like molecular networking, for which you need the MS spectra, but you don't get those from the imaging data. So from this data, we could then create a molecular network, which shows the structural relationship of all the features that you detected in your imaging data. But the structural relationships of these clusters are created using the annotations that come from the LC-IMS data. So this is quite a powerful way how to combine different types of data sets. Now, what I would like to show you is that this actually doesn't have to be difficult and doesn't need to take much time, because I mentioned we now have this wizard, and the wizard creates batch steps for you. So by selecting what type of analysis you want to do, you can create a batch, and then you can just load your files and you can run your batch. And now you can see this is running. It's creating some tasks that are processing. First, detecting the masses, so detecting the individual ions in the different types of scans. Then, can't really see on the screen, but it's now doing the feature detection, so the chromatographic dimension, and then the resolving, assigning the MS scans to individual features, and follow up by the alignment, and also the spectral library search for annotation. And now you can immediately see in your feature list the results of the spectral library annotation for the features that you detected. And we can show the results individually, and then here you can see the alignment of the library spectrum versus your experimental spectrum and the different candidates. So as you can see, this whole processing of this data set that had 6 samples took less than 1 minute with the batch wizard. And in the final few slides, I would like to highlight 2 case studies where we used this ion mobility separation and data processing in MZmine. So first is coming from a postdoc in my lab who's studying paper alkaloids. So these are plant samples. So these plant samples were collected from botanical gardens and analyzed by LC-IMS on a C18 column. And what we noticed is that some of the alkaloids that we are interested in have structures like this. So they have 2 double bonds next to each other. And so they can form cis-trans isomers. And although it's possible to make 4 different cis-trans isomers when you have 2 double bonds, we see only 3 peaks in the chromatography dimension. And I think that makes perfect sense, because if you rotate one of those double bonds, you get very different shape of a molecule, so you get a different retention time. But if you rotate both bonds, then the molecule will actually look quite similar, so it's not going to be separated by the LC. But if we add the ion mobility dimension, we can actually see 4 different signals, so we can resolve all the 4 cis-trans isomers, and we can detect those in the data. So if we do the data processing, then we end up with a feature table like this, where with the blue rectangles, I highlighted those features that have the same retention time, but are separated by the mobility value, or they have very similar mobility value, but they have different retention time. Okay. So what this allowed us to do was to separate all these isomers, and also depending on what instrument you're using, there are different ways how to do MS data collection, but typically the MS data are acquired during the mobility separation. So if you have a peak in the mobility dimension, the blue rectangle now shows where the MSMS data was collected. But in a situation like this, where you have two isomers separated by ion mobility, the MSMS data collection might result in kind of chimeric spectra. And now in MZmine, you can actually tell MZmine to select the MSMS spectra that were collected in the right time. So you can actually do something like this, where you fit the MSMS assignment window to the peak in the mobility dimension. So you only get MSMS signals from that one isomer. Okay. Now the second example is analysis of lipids from mouse spleen. There was data from Patrick Helmer from Münster. And here, as I mentioned before, we use the mobility dimension as an additional descriptor to align different modalities of data. But it doesn't have to be just mass spec and imaging. It can also be like two different types of chromatography. And so here, we use two different chromatography systems. So for one feature in MZmine, you can actually see there are two traces in that chromatogram. And that's because one is coming from helic separation, and the other one is coming from reverse phase separation. So you can cover a wider range of lipids this way. And these were aligned by the mobility values. So we have the same mobility value because it is the same compound. So we can again use the mass and the mobility value to align these data sets, even though the retention times are very different. And then what we can do is we can also include the imaging data. So you can analyze the same samples with imaging. You get the distribution of your compounds, and we can align those with the chromatography data sets using the mobility. And then you end up with a feature list like this, where you have the extracted images and the data from your helic separation. You have the data from your reverse phase separation, and then you have the data from your imaging. Everything is nicely aligned, the same feature. Okay, and this was all regarding ion mobility data processing in MZmine. In case you would like to know more, we have resources. So we have a website about MZmine. We have very extensive documentation pages. We are quite active on Twitter, so you can follow on Twitter what's happening in MZmine. And one of the things that we announce on Twitter is the MZmine Web Cafe. So we do this quite irregularly, but let's say once in 2 months, we have like a Zoom meeting for all the users of MZmine. You can just connect, you can ask any questions, you can tell us about what you're working on, and so on. We're happy to hear from our users. In case you would like to contribute to the development, you can look at our GitHub, or you can also file issues on the GitHub in case you're having some troubles. And to add to that, we also organize workshops where you can learn how to use MZmine hands-on. So there's no space for hands-on training in this kind of session, but these are several days long workshops where we try to teach people how to use MZmine and also GNPS and Sirius and other computational metabolomics tools. We have also already organized a few of these workshops, and next year there should be 2, probably one in Prague and one in Japan. And with that, I have to acknowledge the community around MZmine. There are some names that I mentioned, Robin and Stefan, also Ansgar and Shusha Duzlab, Daniel Petra. These are the people that have contributed a lot, but also many, many other developers. You can see the logos of some of the institutions on the right side. So there are many people who contributed over time. And also funding from the Czech Science Foundation, some funding from Google through the Google Summer of Code program, and also the Cost Action Epilipidnet, which has been very supportive. So thank you all for attention.
Are there any questions for Tomáš? No? Okay, I have a quick one. So in your experiment where you were combining the multimodal data, What percentage of features between the two data sets were actually able to be merged?
That's a good question. I honestly don't know off the top of my head because I have to admit I don't work with ion mobility data myself. But as long as you have the mobility values, right, then you can always align the features. I think the biggest problem could be if you have some ions where the mobility value is not different enough, right? So then those could get messed up during the alignment process. But so far, at least based on the examples that we show in the paper, it seems to be quite powerful because the mobility dimension now is, like the resolution of the mobility dimension with the current instruments like the timsTOF is actually pretty good. Yeah. But yeah, regarding the percentage, I would have to check.
I'm just curious because sometimes between MALDI and LC, you see very different adduct types formed, and that could be another complication between merging them because the drift times would be quite different.
So I have a question regarding the first step, which is when you are identifying the peaks in the mass dimension. Do you do it for all the mass? I mean, in the IMS experiment, you collect tens of time of flight, or mass spectra basically, from time of flight. Mm-hmm. Do you do that detection for every single spectra you come, or do you do some sort of summing or across the IMS peak? Because the peak goes from baseline, go up and then go down.
Are you talking about the very first step, the mass detection, essentially? Yeah. Yeah. So we do that for all the scans because it actually reduces the size of your data. So in the spectrum, you have a lot of data. If you collect profile spectra, such as we do with the Orbitrap, then obviously you have continuous signals. It's a lot of values. And you can replace those by essentially just the centroids, and you can also remove noise. In case of centroid data, which is typically with the IMS dimension, we collect centroid data, then you only reduce the noise, but you still reduce the amount of data. And you can do this already when you're importing the raw files. So you can, in the import module, you can select to already denoise the values, which then saves quite a bit of memory during the processing.
Okay, well, if that's all, then we can thank Tomáš one more time.
Thank you.
Have a quick break while we switch over the computer for the next presentation. Just give us a couple minutes.
What's that?
I also have USB-C.
Might be able to just plug it into it. God.
Give me a sec, I'll mute the microphone for a second.
Oh.
You have an adapter for your...
This might... Yes. Yeah, it has HDMI. So.
Very straightforward, eh? Just be mindful about the microphone. You can feel free to adjust it.
Okay.
Okay? That's the light. That's okay. Just making sure. And that should, if you don't mind. I have.
So that's just one side, but we plan to do it interactive. Yeah.
Okay. So they plan on doing interactive. That shouldn't be an issue, right?
Anything that's showing on your laptop showing on the screen.
It's going to show on the screen.
Okay.
Is that all fine with you?
Yeah, that's fine.
Okay. Awesome. Yeah, just be mindful, like I said, of the microphone. You won't have any timer on. Oh, they do have a timer. Hey, maybe sometime you can make bigger. Do you require a timer at all?
That would be a nice notification.
Yeah? Okay. We won't decide or send out.
Okay. Okay. I think she has.
I think, yeah, Kelly can help us out. Yeah. Let's see. Oh, it might actually. Can we do it here? I don't know. It's kind of. Yeah, it's kind of. Does it go?
Cable's plenty long, so if you want to move it to the table.
Yeah, I think that.
It's like 25 feet long. There's mics on it, so you guys can sit beside each other.
We can leave it on the other. Oh wait, where's the?
All right,
Yeah.
our next presentation
In case
is
they
going to
wanna
be from Christine Chang and Jess Bade from PNNL, where they're data scientists, and they're going to present on the new program that they've worked on, DEIMoS, that allows you to do peak detecting in alignment with mobility data.
Awesome. Okay. Wait, this.
Can everyone hear us? I think so.
Yeah. Okay. Okay. So hi, my name is Christine, and this is Jess, and we're both developers of DEIMoS, which is a software tool for multidimensional mass spec processing. We've actually prepared an interactive Jupyter notebook for this workshop, so if you want to follow along, we have the notebook and some included example files available at the Zenodo link. You can download all of those and follow along, but we'll also be running through everything as well. We do have a real world data file or a set of data files as our examples, so there may be some difficulty downloading such a large file. So, but if you're able to download from our Zenodo link, then feel free. Otherwise, feel free to download and follow the notebook yourself later.
Additionally, there's also just an HTML file that you can view the input and output more up close, since you'll be viewing code on a screen, and that won't require downloading the mass spec files. And if you are searching it on Zenodo, it's DEIMoS Metabolomics 2023 workshop.
So this is the Zenodo site. If you see that, you've reached the correct link. Okay, so I guess we can get started with the demo itself. Switch to the notebook. Okay. Can everyone see the screen well? Should we increase the text size? Maybe a little. Maybe like that. Awesome. So DEIMoS is the program we'll be discussing today, and it stands for Data Extraction for Integrated Mass Spectrometry. And the development for DEIMoS really arose out of an existing need that we encountered while processing our own data at the Pacific Northwest National Lab, coming off of our instrument of LC-IMS, MSMS data. And we wanted sort of a software that was capable of handling IMS data that could be integrated easily into computational programmatic workflows and do extreme high throughput data processing of hundreds or thousands of data files at once. And the existing tools at the time really struggled, at least with the IMS portion of the data. And so that's where a lot of our development started as well. So DEIMoS has always been developed with an eye towards IMS data, but more broadly speaking, has been developed to be a platform agnostic, and the code is actually written to make DEIMoS flexible so that in the future, when new instrument technologies are developed with their own data processing needs, it's designed to where once the general importing of the data and data processing algorithms are all designed to be very flexible and not specific towards a certain modality. And so we're hoping that this opens up DEIMoS for future instrument development and makes this future-proof or friendlier towards advancements in mass spec technologies. And so we've sort of included in our notebook links to the publication for DEIMoS. DEIMoS is also fully open source and publicly available, and the link to the GitHub is there at the PNNL Pacific Northwest National Lab GitHub repository. And we also have pretty extensive documentation that was prepared by our lead developer, Sean Colby, who isn't here today, but that's available through Read the Docs. And so we've just included some setup instructions for those following along currently or in the future at home. But essentially, you can download DEIMoS via Git, and the instructions are included there. And for those of you who aren't familiar with the Python or Anaconda ecosystems, the easiest way to install DEIMoS is via Conda. And to do so, we've included an environment YML file, which includes all of the dependencies for DEIMoS. And so you can simply install all of those dependencies via Conda, and that'll produce the needed environment. Once you have the environment downloaded, you can Conda activate that environment. And from there, specifically for this tutorial, if you don't already have Jupyter, we've included that here as part of the instructions. And then once you have all the dependencies downloaded, you can then install DEIMoS via the GitHub repository, which presumably you've already cloned via GitHub from the instructions as well. And the official documentation does go into a little bit more detail on the installation. If there are further questions, we've kind of simplified the instructions for the purpose of the workshop. And then once all of those steps have been completed, you can launch this notebook via Jupyter. And so here you can just access the directory containing the notebook and then start Jupyter Lab. This notebook is starting from that point, assuming you have followed all those steps already and are ready to now process your LC-IMS data. So as Tomáš talked about previously, there are multiple data types for LC-IMS, MS data, and different instruments from different manufacturers can result in different data types. DEIMoS assumes open source input-output data formats, and so DEIMoS accepts as input the open source MZML format, which was discussed in the previous presentation as well. If you have instrument data from, say, Agilent or Bruker, you can convert that into the MZML format using a utility such as ProteoWizard. They have an MS Convert utility, so that's alluded to in the documentation. And there's a link to ProteoWizard as well in this notebook. But to start, we're starting with data that's already been converted into that MZML format for the purposes of the demo. And so here, I'll just start by running this notebook. And so if you download directly from Zenodo, the data will come included in this data folder. And here I'm just pulling the data files that we have using Python globbing. And so just grabbing all of the data files here. Here we have 2 included, and I'm printing an example file name from one of them. So this actually came off of one of our instruments at the laboratory. And I can use DEIMoS to look at your MZML file. So we'll import DEIMoS and use DEIMoS to read the accessions in the MZML file. And for those of you who aren't familiar with the MZML format, it's essentially stored in an XML-like format where it shows you all the different fields that are accessible in that data. And so here you can see the different fields that you can access, including the MS level, the ion current, et cetera. Here, this is LC-IMS, MS data. And so we are interested in grabbing the drift time and retention time dimensions. I think M over Z is grabbed by default by DEIMoS. And so here, those happen to correspond to the scan start time is the LC retention time dimension, and the ion mobility drift time dimension has the succession. And so to load that data, we'll grab that file and specify those accessions that we want here. And so I'll just load these data files, or this data file here. The data will contain both MS1 and MS2 data. And so I guess I'll just wait a second for this to load. And then once the data has loaded, we also wanted to demonstrate DEIMoS allows you to save data in different data formats, but we like the open source HDF5 data format typically. And so to load in DEIMoS, you use DEIMoS.load. To save, you use DEIMoS.save. And so we'll just save the example MS1 and MS2 data. And that might take a second to access the data file because it's quite a large one. I think it's about 3.5 gigabytes. So.
Additionally, when running a Jupyter notebook, it's off a single core in your machine. There's no like multi-threading with a Jupyter notebook, so processing would look a bit different in like a high throughput workflow.
Mm-hmm. Okay. Any questions so far? Should we just wait for this? I guess we can keep going. I guess we can kind of show what it looks like while it's loading. Yeah. Yeah, so while the notebook is running, we'll just show what the output should look like. Essentially, you can see the data will load into a pandas data frame, and so you can read the data, in tabular format, and you can see there are quite a bit many rows. So that's what's taking the notebook a while. Mm-hmm. I think it's still going. It's okay.
Can I ask a question while we wait?
Yeah.
So I know there was a new paper that came out recently from IVET about the EMZA.
Yes.
So how does that how would that sort of be implemented into DEIMoS?
So we do work closely with IVET, and I think we don't have inbuilt compatibility with MZA format yet, but that's we are aware of MZA, and that'll be probably implemented in a future version of DEIMoS. For now, we're mostly assuming MZML as the standard format, since that's more commonly used in the field. Okay. Okay. While the data is loading, and I guess we'll run the saving cell as well, I'll just move on to discussion of peak picking. And so to identify features in your data, DEIMoS can perform peak detection. And we went through a few iterations of different peak picking algorithms, but the algorithm that we currently settled on as the default for DEIMoS is using persistent homology as the basis for signal detection. And so we've seen with persistent homology, it does a good job detecting, the features in the data versus other approaches such as, local maxima filters that we've used, as well as scaling really well to large multidimensional data sets, such as the ones we process at the lab. And so, for instance, our peak picking algorithm on this data file, which you can see is quite large and takes a while to load, takes a matter of seconds. And so we're hoping we'll demonstrate that in a bit once the data has loaded. You want to add something, Jess?
For those of you who have read the DEIMoS paper, our initial approach to peak picking did involve local maxima. And in addition to the improvements Christine mentioned, additionally, by switching to persistent homology, we no longer have to partition the data, which removes potential influence from edge effects of, depending on how you were to cut the data to process it in its native dimensions. And so we have removed that from the current peak picking approach.
Yep. Okay. So I guess we can cover the peak picking in the notebook. Should I just run? I'll just run a bunch of these, and then we'll go through the notebook. Okay, we'll just run a few of the cells and then go through the notebook and the HTML just for time reasons, but I'll just run a few of these. Okay, up to pre-processing. So, in the previous section, we demonstrated loading and saving data, and so you can load one of the data files that was saved that corresponds to the MS1. And when data is in the HDF5 format, you can specify a key to access which, as part of which data you're accessing. You can see that in the save section that you save your data under a specific key. So here we're loading the MS1 data, so the key is MS1, as you can see in this above cell up here. And so we just specify that here. I'm cutting down the columns so you can see from this raw data that there's this scan ID, retention time, drift time, et cetera. I'm going to load specifically the M over Z, drift time, retention time, and intensity. And then from there, of course, when you load your data, many spectroscopists like to visualize their data as in total chromatograms or mobilograms, just so you can get a broad picture of what your data look like. And so here we're using DEIMoS to produce those total ion slices of the data using this DEIMoS.collapse utility. What that does is collapse all the data into the specified dimension. And so here, I guess I'll make it a little bigger. Here we're collapsing in the retention time dimension for that chromatogram and just using DEIMoS' plotting utilities to plot what that looks like here. And so, and then we can do the same thing in the drift time dimension. We've collapsed that in the drift time dimension and are now producing that total ion mobilogram. So you can have a pretty high-level picture of your data. Alternatively, DEIMoS can also plot a 2D representation of your data to visualize what we've shown above. And so here, I collapsed in the drift time and retention time dimensions, and then we're plotting that 2D data using this grid plot. And so it's sort of more of a heat map type of plot, but you can see the drift time and retention time dimensions. And it looks like the notebook has actually run through those cells, so we're caught up now. Great. So now that we've visualized our data and loaded and saved it, before proceeding to peak detection, first we perform smoothing. And so we've here included smoothing parameters that are we've by default determined using lots of tune mix data and iterating through different parameter sets. But of course, those can vary based on your own data, and so that can be adjusted to suit the user needs. And so we do that. We build our factors from the raw data. We apply nominal threshold just so that this is a very large piece of data, and so we're just applying a very small threshold, intensity threshold of 500, just to reduce that a little bit for the future steps, and then we'll apply the smoothing to smooth the MS1 data. And something we'll note here about the parameter sets is we did test a variety of different parameter sets for smoothing, and we found in general that increasing the number of iterations for smoothing and keeping a small smooth radius tended to perform best, but does eat up more computational resources, and so there's sort of that trade-off there. And then now that we have our data smoothed, we perform peak detection. And so peak detection performs peak picking per dimension, and you supply parameters per dimension for that purpose. So you see in the radius parameter, you can specify the peak detection radius per corresponding dimension. And so these parameters are dependent on the resolution of your data, and again, we've predetermined these based on existing data sets on our end, but those can be adjusted based on the resolution of your instrument and data as well. So now that we have our MS1 data, smooth, we'll perform peak detection. There we go. Oh. Okay. And so peak detection, we included a little timing notebook magic here to demonstrate the timing here, and you can see that the peak detection is fairly fast. It takes a matter of seconds. And then just to visualize one of the features, we're just including another plot that DEIMoS can generate is this multi-panel plot where you can visualize in each dimension what a feature looks like. So here we've pulled an example feature from that peak picked list and visualizing what that looks in 3D. And so there are the 2D representations on the top row, and then the 1D representations with the intensity dimension as the y-axis on the bottom. And so you can see this is a very intense feature. Looks, reasonably peak-like in all dimensions. So now what everyone is interested in this workshop for ion mobility, CCS calibration. And so many of you are probably familiar with this equation here. The Mason-Champ equation can be reformulated into approximately linear format based on these included parameters. And so if you have, you can calibrate your CCS for your instrument based on a mixture of ions with known CCS values, and that's the process known as calibration. And so DEIMoS is designed to accept two different formulations for CCS calibration. And so in the first method that DEIMoS can perform is direct calibration of CCS from a data file itself, as long as that data file contains the tune mix ions. DEIMoS can also perform manual calibration. For instance, if you run CCS calibration on your instrument and you just get some type of PDF output, I think Agilent does this, where you run your tune mix and it gives you an output for that day of what the CCS calibration parameters look like. It can also perform manual calibration where you can feed those numbers into DEIMoS and it'll accept those into the calibration. And so we'll just demonstrate those here. Here we've included, as part of our example data, an actual tune file from the same day. And so this might take a while because we're loading the data as we did previously from MZml. We use this DEIMoS.calibration.tuneMix function and load in that tune file data, and we're looking specifically for certain tune mix ions. This is just from the Agilent tune mix. Specify the buffer mass, here it's nitrogen gas, and include a tolerance in each dimension where it'll search, DEIMoS will search for those tune mix ion peaks and then perform the calibration. And you can see the results from that calibration are here. The R squared is very high, and then you can calibrate the CCS for your data based on the results from this calibration. And so I've done that here to add our CCS to our original data. There's also this manual calibration, like I mentioned before, that's just done through this DEIMoS calibration.calibrate CCS. And again, you can feed in that list of expected ions and known CCS, and it'll save those calibration parameters as well, and you can apply those as needed. But here, since we have a tune file, I'm not going to apply that to our data. Okay. And then once you have your CCS calibrated, the data that we've included here also has fragmentation data, MSMS. And so we'll perform the same thresholding, smoothing, and peak picking on the MS2 data here. And while that runs, do you want to go through the offset part?
Sure, yeah. And then for the next step, we'll be talking about a drift time offset phenomena and how we correct for it in our drift time values. And so...
Wait, should I run? Maybe I'll run the cells while you talk.
Mm-hmm.
Yeah.
And so a phenomena we noticed is that there is a non-uniform drift time of fragments, different from the precursor drift time. And we've noticed that this occurs as the ions pass through the transfer cell, and there's an electric ionization or electric potential difference that causes the fragment drift times to arrive sooner than would be expected. And so we've tested and introduced an equation that is a function of the precursor mass to charge, the fragment mass to charge, the collision energy, as well as the neutral loss. And we apply this correction, which we'll show in the code below, to correct the fragment drift times, which improves our deconvolution.
Mm-hmm. Oh, yeah. So I ran all of them through decon, so if you wanted to talk about it. Yeah, so since there's an offset between the drift times for the precursor and fragments, you can use DEIMoS to apply that offset, and so you can better associate the MSMS fragments with the precursor. And so we have included, so the way that this is designed to be handled in DEIMoS is essentially you can specify your offset correction model. We've included an offset correction model that we've determined based on the analysis of our data. And so we found a relationship that, as Jess mentioned, includes the overseas drift times and then the this collision energy as well. And so here we're just including that offset correction model, but you can specify your own. And then once you apply that offset, now you can better associate those fragments with the precursor with the precursor peak, and then from there, DEIMoS can then perform deconvolution, the process of associating those MS fragments to the precursor and producing the MS2 spectrum. And so the deconvolution step does take a bit of time, and so I'll run through the example on the HTML while the notebook is running. But essentially, what we do here is we apply a threshold so that we are looking at some of the more intense features that were identified via peak picking. And then once that's performed, we use this MS2 deconvolution model, including the MS1 peaks, the MS1 raw data, the MS2 peaks, and the MS2 raw data. And then we construct pairs of MS1 features associated to those MS2 fragments. And you can, again, specify your offset correction model, so it'll apply that offset correction to your fragments. And what you get as output is this table of MS1, your precursors, and then the MS2 fragments, and it contains information in every dimension. And also this column here at the end, wait, where's the score? Oh wait, the score is the next one. Yeah, so it'll apply the offset correction model, and then you can deconvolve the MS1, MS2 features, and then you can, here we've sort of grouped together all of those MS2 features by the precursor ion here. And then each of those precursor ions then has an associated score in drift time with it, which corresponds to how well it's associated with the precursor. And so if your fragment has a very high drift time score, then it's associated to that precursor with a very high confidence. Here we're just showing an example deconvolved result for one of the precursors. The associated fragments are shown here in this MS2 spectrum, and we've color encoded these peaks by the drift time score. And so we've applied a threshold of 0.9 to those scores, where 1.0 is perfect association, so 0.9 is a fairly high drift time score, and we can use that to filter out some fragments that may not be as associated with the precursor, and those are shown in the gray on this plot. And so you can see that this fragment here at about 575 MRZ scores below that threshold, but the rest of the fragments here are very highly associated with this precursor based on our deconvolution algorithm. Okay, I guess we could take a quick break. Are there any questions before we move on? Because we're going to go into sort of a directed application of DEIMoS. Okay. If not, we'll move on. So, of course, what everyone is interested in is the actual application of their software to their data. And so one application that we have had in our own work is, the identification of compounds from mass spec data. And so in real-world applications, typically in mass spec workflows, you might have a list of unknown compounds that you're trying to identify in your data. And so DEIMoS has some inbuilt utilities to aid in this process. And so utilities for data analysis and for identification of features in your data. And so in this application section, we're demonstrating that with an included list of standards that are spiked into our data. So the composition of our data is actually a plasma sample, so a real plasma sample that's been spiked with various internal standards. And we have a list of those standards in this internal standards file, and that includes information such as compound identifiers, and also information on the adduct mass, M/Z, and for some of them, the expected CCS. So here we're going to demonstrate how to identify some compounds in this list, in the included data via DEIMoS. And so here, I'm just loading the target list here using pandas, and then we'll only consider a subset, the subset of targets that have CCS data for the purposes of this demo. And so just to demonstrate, how to search for specific compounds, let's look at a random compound on the list. And so here, let's just look at the 22nd compound on this list that happens to correspond to this compound called phenylacetylglutamine. And so to search for a feature, you probably want to look within a certain M/Z tolerance. And so here, we're just going to use a tolerance of 20 ppm and convert that from relative to an absolute tolerance. And then you can use this DEIMoS.locate function to look at our, in this case, our deconvolved MS1, MS2 data. We're going to search for that feature in the MS1 by that target M/Z and only search within that search radius of 20 ppm. And what we get here is this a list of matched features. So you can see this feature in MS1 at 265.11 is fairly close to our expected ACT M/Z here. And then you can see that these are each row corresponds to a fragment peak in MS2. So we're just going to reproduce some of the steps we saw above. We want to look at this feature and its associated chromatogram in 2D and 1D space. So what does this look like? Sometimes chemists can immediately tell when a feature is junk just by looking at its peak here. But these look like fairly well-resolved peaks of a pretty high intensity in each dimension. And of course, we can plot the extracted ion representations as well. Here, we're sort of reproducing the steps in the total ion, except we're filtering in that M/Z space to restrict this to only the signal of interest. And so the dotted line here represents the feature that was found using DEIMoS' locate function. And so you can see that corresponds fairly well to the maxima on both of these extracted ion plots. And of course, we'll plot the MS2 for the extracted feature. And again, I've included color encoding by the drift time score here that's determined by deconvolution. And so we get this fragmentation pattern. And it's not shown here, and DEIMoS doesn't include scoring utilities, but actually we looked in MONA for this, and the fragmentation spectrum for phenylacetylglutamine corresponds pretty well to this fragmentation spectrum. And so you can draw in scoring utilities such as cosine scoring and score those against existing databases. And then here, DEIMoS also includes the ability to save data into the MSP format in case, for computational workflows where you extract many MS2 spectra, you may want to save that into a standard database format. And so many of you are likely familiar with MSP. And so you can just apply those parameters into the DEIMoS save function, and it'll save as MSP automatically. And so here, we've included as part of the included data files an example MSP, and so this corresponds to that signal that to that feature that we demonstrated in this workbook, and so it'll save, in MSP format, which looks approximately like this. There are also further capabilities of DEIMoS. DEIMoS is you can perform alignment using DEIMoS. And DEIMoS is designed to be integrated into larger workflows, and we've actually developed several workflows at PNNL that integrate DEIMoS into, for instance, a larger snake-make workflow where you can perform a variety of those functionalities. It's automatic snake-make and Nextflow, for instance, can scale very well to HPC resources or cloud. And so if you have very high-throughput data processing needs, DEIMoS is designed to integrate fairly well into those. And I think we might be should we go into alignment? I feel like it might take a while. How are we doing on
How
time?
are we on time?
Oh, okay. Okay.
Yeah, and then I guess do you want to just...
Yeah, so we included an example of cross-sample alignment in DEIMoS, so we can go through that example.
Oh.
Oh,
Yeah, if you're interested in that, you can look through the code
Yeah.
in the document.
yeah, it's in the
sure.
notebook, but we don't have to go through it specifically, and it's also in the documentation.
Any questions?
Okay, thank you. So I will say my students who have very different coding abilities have figured out how to use DEIMoS and really like it. So it might seem a little complicated at first, but I love that you guys made the Jupyter Notebook for us to try out, and there are some really good resources, and they're also very responsive with questions and stuff too. So it's a cool one. I'm excited to see what happens with it. Are there any questions? No? Okay, so then quickly in the last few minutes, thank you guys so much for doing that.
Yeah, thank you.
Okay, so in the last few minutes, I have some slides to show you about CCS prediction and places for finding CCS values. So a nice summary of kind of like wrapping up the final part of your experiment where you've got to start working on those identifications and figuring out what you've found. Awesome. Okay. So, you've done ion mobility, and you have CCS values that you want to compare and see if you've actually, matched up to some stuff. There's a lot of places now that you can find CCS values. So this table I made sort of summarizes the big ones that I'm aware of that have like the most values for small molecules, so metabolites and lipids. The big one is the CCS compendium from the McLean lab at Vanderbilt University. This is exclusively drift time ion mobility CCS values. Aaron Baker and also PNNL have their own databases as well that you can go in and look at. Those will also be drift tube CCS values. And then we start getting into some resources that have a bit of a mixture of values. So PubChem, if you maybe have missed it. Now, if you go in and look at a metabolite and scroll down a bit, they will actually have CCS values for quite a lot of stuff in there. And so those are a mixture of drift tube CCS values and potentially calibrated values from traveling wave and trapped ion mobility. And what's really nice is when you look at the entry, it will tell you very specifically how that CCS value was determined. So it will say drift tube ion mobility, or it will say traveling wave calibrated with polyalanine or something along those lines. So it's very nice and clear. Then a couple of sources have a mixture of all of them, and this is because they were originally sort of developed and curated as the training set for machine learning-based CCS prediction. And so some examples of those would be CCS-based from the Shu Lab at University of Washington. The website is ccsbase.net, so you can go on there and look at it. They've used drift tube values, traveling wave values to develop their prediction algorithm, and so that's all in one place. The Drew Lab from Chinese Academy of Sciences also has a huge like collection of drift tube and drift tube, probably, I would say they're probably mostly trapped ion mobility, calibrated values, and predicted values that you can go in and search as well. And so you can search this in all sorts of ways. You can put in formulas and come up with predictions. You can put it in a mass, get back some potential identifications. You could put in a CCS value, put in some identifications. It's a really great, great tool. And so that one's called All CCS. And you may have seen there have been some sort of previous versions of this, Met CCS and Lipid CCS. I think they're all sort of combined now together into this All CCS tool. And lastly, HMDB also has collision cross-section values. They're going to be a mixture of drift tube values and predicted values. And the predicted ones you can actually pick from a couple of different prediction algorithms that you're interested in looking for. So it will give you the prediction from all CCS, deep CCS, or the dark chem CCS prediction algorithm. So I wanted to make sure we got that so you can get your picture and have it on hand. So really, the CCS prediction method has become kind of the go-to way for doing this. It's just too complicated to get drift tube measurements for all of the potential metabolites or lipids that you might find in biological samples. What that really means is that, like, you ideally would like to have a standard, or you need to have done like a very thorough identification so that you know that this is actually the right identification before you start putting those values out there. And with the growth of the drift tube ion mobility instruments, right, we do now have a collection that's a pretty reasonable amount of CCS values for these molecules. And so they could be used to train prediction algorithms that will help us sort of more quickly come up with CCS values for things that we can't necessarily measure directly in the lab. And so the idea here is that you take your development set, you have some set of compounds, you know their structures, you know all of this information about them, and you have their CCS values. And you let machine learning figure out what are sort of the key molecular descriptors that will sort of predict a CCS value. And so the Zhu lab really was the first to do this, as far as I can remember, with this Met CCS program. So they really went through a very exhaustive process of collecting drift tube ion mobility data, training the algorithm, testing the algorithm, and showing that it works really quite nicely. So that in the end, what you all now are able to go do is put in some information about a molecule you're interested in, and it will predict a CCS value. And so it's got quite a large collection for metabolites, 35,000. One thing to note are what sort of adducts are included in the database, because this can have some influence on the quality of the answers that you get back. So if your adduct type is not really well represented in the training set, there might be some error there. So this is an online utility. Another one, this is from Facundo Fernandez's group at Georgia Tech, which is something that you could have more on your own computer. This is CCS Predictor 2.0, which is set up in a Jupyter notebook like you just saw Jess and Christine run, and then also as a Google Collaboratory page, which is something new to me. I haven't heard of it before. But it uses a very similar strategy, right? So you've got some kind of training set. You are looking to identify like which molecular descriptors are going to be the best use for predicting the CCS values. And what's nice about their workflow is that, say you get a candidate structure from something else like MSMS, you can use that candidate structure to get the descriptors and come up with a predicted CCS value. Now, the two sort of methods we had talked about before have sort of like one model that was developed. So MetCCS has one model that was developed for lipids. I think there's now another model, for metabolites. Sometimes you don't necessarily know if what you have is going to look or be more like a metabolite or a lipid or some other kind of molecule. So another way of doing this is using unsupervised clustering. And so in this method that's implemented in CCS base, they're using an unsupervised clustering method where it's going to look at the molecular descriptors for each of these features and assign a particular prediction method to that feature based on where it falls in the clustering. And so you can see on this example of what they've shown, the coloring here for these different clusters is based on the identity of the molecule. So there's lipids, small molecules, carbohydrates, I think is the other one, and peptides. And so you can see that they fall in very different places of a mass to charge versus CCS plot. And they also are in different places within this clustering. So there's models that are developed specifically for predicting CCS values for lipids, a different model for metabolites, small molecules, so on. So if you're really uncertain about what you have, this might be a resource to try. Now there's going to be a bit of variability amongst these different prediction algorithms. I think for the most part they do decently well, and they're a great resource because you can't reasonably go out and buy a standard of everything and find someone with a drift tube to run it for you to check experimentally. And so this is a great resource, but it's just important to recognize that the algorithms and these sort of machine learning methods are really going to perform best for things that they were trained on. And so it's important if you're working on something that's maybe an unusual model system that might not be well annotated. Like I know I do stuff with bacteria. Their metabolites, their lipids may not be well represented in these training sets, and sometimes the adducts might not be there as well. So you might see higher errors in that sense. Just do a little bit of investigating yourself to make sure that you're, picking something that's reasonable. The last thing I want to touch on is this decision that you would have to make about actually selecting a CCS threshold. So just like we've all become accustomed to saying when we're doing accurate mass filtering, you would say, okay, I use a 5 ppm cutoff. So we're going to say anything above 5 ppm I don't really want to consider, or maybe it's 10 for you. You need to have a similar threshold for doing these sort of CCS matching, and there really, I think, has not become a good sort of like consensus yet on what that should be. And so it's still a little bit of a gray area. So things like for quite a while, 2% was sort of the number that people were using. But this example from Facundo's group shows really nicely the trade-off here. So if you're using CCS value as a way to filter out candidate identifications, to some extent, if you become too stringent with that CCS cutoff, you'll potentially be missing sort of like the true or correct identification. You'd be filtering out a lot of wrong identifications, but you might also lose the correct one. And it can change quite a bit for different types of molecules. So lipids might be slightly different than metabolites, and so it's important to sort of test this for yourself to see how it works. In this particular example, they're using a 3% cutoff. And so you can see that the percent error between the sort of true identification or the true CCS value and what the model has predicted for a large set of metabolites all tends to be kind of skewed to a lower mass range. And so there's just some trends that you might pick up on when you're looking at these. Okay, so finally, just to sort of summarize where we went today and what we covered. So there's a bunch of software programs that are available now, some really good ones that are out there for people to use to incorporate ion mobility. They cover the whole gambit from peak picking to alignment to identifications. You can use ion mobility as a way to filter those identifications, to drop down that list so you're not swimming in potential identifications there. You can use it for cleaning up this tandem MS spectra, and there are now many resources for finding CCS values. So they're both experimental values and predicted values. And so just keep in mind here that the sort of key decision processes that you have to make is to select one of these databases that is sort of the best potential representation of the types of molecules that you might see in your experiment, including the adduct types, and to sort of balance this CCS threshold properly so that you don't cut out too many things while simultaneously keeping the right identifications there. And so at the end, I think we have maybe like 10 minutes before the next one actually starts, but we probably should sort of clear out so they can come in and get set up. If you have any questions, I'll hang around and I'm happy to take questions. You can also email me or find me on Twitter, and I'm sure the others who've presented are the same way. But thank you so much for attending, and I hope you learned a lot and feel like you've got some good go-tos to get started with ion mobility in your metabolomics workflow.